Welcome back to the After School Project. In part 1 of this nucleic acids series, we introduced nucleic acids with a specific focus on the DNA molecule and DNA replication. In part 2, we look at the RNA molecule with a specific focus on protein synthesis, but first, here are some key differences between DNA and RNA. DNA exists as a double helix, while RNA exists as a single strand. DNA contains deoxyribose sugar, while RNA contains ribose sugar. The nitrogenous bases found in DNA are cytosine, guanine, adenine, and thymine. The nitrogenous bases found in an RNA molecule are cytosine, guanine, adenine, but thymine is replaced with uracil. There are 3 types of RNA molecules. Messenger RNA that is found in the nucleoplasm. This is formed by forming a complementary strand on a DNA molecule. It carries a genetic code from the DNA to the ribosomes where protein synthesis occurs. There's also transfer RNA that is found in the cytoplasm. It is a single strand that folds back on itself to form loops. On one of the loops are 3 exposed nitrogenous bases, often referred to as an anticodon. It picks up amino acids during protein synthesis, and the amino acid it carries depends on the anticodon on the transfer RNA. We also have ribosomal RNA that occurs as a single strand and plays a role in protein synthesis. The process of protein synthesis happens in 2 parts. First, the transcription of the DNA molecule, and secondly, the translation of the messenger RNA into proteins. It is important to understand that the DNA molecule carries genes which are a sequence of 3 DNA nucleotides that control the formation of specific proteins. But the DNA molecule cannot travel outside the nucleus, so we need to copy its coded message to the ribosomes where proteins are formed. In the process of transcription, a section of the DNA molecule unzips and unwinds. Free-floating RNA nucleotides form a complementary strand only on one side of the DNA strand, with cytosine always pairing with guanine and adenine pairing with uracil. A single messenger RNA strand is formed. The messenger RNA, once formed, moves away from the DNA strand, and the DNA strand joins again and twists to form a double helix again. The messenger RNA leaves the nucleus via the pores in the nuclear membrane, taking the code from the DNA to the cytoplasm. In the process of translation, the messenger RNA formed in the nucleus attaches to a ribosome. A transfer RNA molecule carries amino acids, which are the building blocks of proteins, to the ribosomes. The anticodons, or exposed nitrogenous bases of the transfer RNA, will find complementary messenger RNA codons and pair with them, with cytosine always pairing with guanine and adenine pairing with uracil. As soon as the transfer RNA places an amino acid on a ribosome, it breaks away from the messenger RNA and becomes ready to pick up another amino acid of the same kind. When 50 or more amino acids are joined in sequence, they form a protein.